on Catalyst. Honeybees. Australia is a safe haven for them, but with the threat of parasites, pesticides and disease, how long can we keep it that way? Quenching your thirst, do we really need 8 glasses of water a day? And what distracts drivers more? Hands-free, handheld or passengers?
There's one animal on the planet that can make or break our food supply. Even the shape of our food depends on it.
Well, there's 5 sectors in apple. If they're not pollinated evenly, then you don't get a perfectly round apple.
We rely on bees to pollinate most of our food crops, and one species in particular.
Of about 1,500 species of bees that live in Australia, this is the most recognisable and the one we take most for granted. The European honeybee, first introduced nearly 200 years ago. They've been here long enough to be considered practically native.
But elsewhere, the honeybee world is in dire peril. Each year for the last decade, about 1/3 of their colonies have died. It's a mystery known as colony collapse disorder, a perfect storm of pesticides, habitat loss, diseases, and a vampire parasite called Varroa.
It's multiple effects. It's not just pesticides and it's not just Farrell.
This is a story from the front line. Farmers, beekeepers, scientists and honey lovers determined to secure a future for bees and for us. When it comes to honeybees, Australia really is the lucky country. We're the last safe haven because the Varroa mite isn't here yet.
Yeah, the only continent in the world that hasn't got it.
Well, that's incredible.
Yeah.
Is that just luck or good management?
Luck.
At a cost of only a couple of $100,000 each year, hives like these stand guard around our major ports to detect invasions of exotic bees and pests. Leatherwood honey producer Peter Norris manages the Sentinel hives in Hobart. He dreads an invasion by Varroa.
We don't want it here. We should do everything we can to keep it out.
Varroa is a genus of mites that suck the blood of bees and their larvae, spreading viral diseases. Compared to the size of its host, Varroa is one of the largest parasites on the planet. This worker bee in the UK has a mite attached. You can see how agitated the bee is, which prevents it doing the jobs that keep a hive healthy. Peter has seen the Varroa invasion before.
Well, I've had some experience. I was in the UK when they discovered it there. I'd been keeping bees for quite a few years there, got out to about 150 hives, and the year that they discovered Varroa, I went down to 25. We lost 80% of the bees in the southeast of England.
In a year?
In that one season, yeah.
What would you expect to see if you had Varroa in this hive?
You would see adult bees with very deformed abdomens, deformed wings, not capable of flight. Because of that, the food supplies in the hive diminish very rapidly.
What is it about the bees?
That devastating prospect could be entering a port right now. Department of Agriculture officers board ships to search for foreign insects, particularly Asian honeybees, the natural host of Varroa mites.
A ship could be docked in an overseas port and the swarm will come and just by accident land on a ship or some cargo. And they will usually hide under a container or under beams. Not every ship is inspected, but there's risk profiling that's done on different ships.
It just takes one bee in one container, in one ship, in any port for Varroa mite to arrive in Australia. And that's the challenge for Australian biosecurity, to stop that stowaway bee escaping in the first place.
Foreign swarms are found and destroyed up to a dozen times a year. In 2011, a swarm of Asian honeybees brought their own unwelcome passengers.
We discovered the swarm had more than 150 Varroa mites associated with it.
So a good one to spot.
It was really important because if it had absconded, then the Varroa mites could have got away.
However, we actually import queen bees from Varroa-infected countries in the hope of breeding their genetic defences into our bees.
If we ever do get Varroa, hopefully some of the queens that are now being imported will have resistance to Varroa, so we'll be one step ahead in the battle against this dreadful parasite.
Ross Rickard runs 3 months of quarantine checks on each import to ensure that not only are the bees clean, but their packaging as well. It's not without risk, and for safety's sake, the bees that arrive here never leave the lab. The queen spends the rest of her days laying eggs in a secure hive. Varroa mites don't just suck blood, they use the hive to lay their own eggs.
When the queen starts laying eggs, we examine that brood and make sure that there's no mites in there.
Beekeepers can dose their hives with chemicals to control Varroa, but wild or feral bees don't get that help. These bees have left the hive to strike out on their own. Unlike the rest of the world, Australia's still an easy place for honeybees to survive in the wild.
There's a lot more feral colonies in any environment than most people realise.
That's not the case in countries with Varroa, where honeybees can only thrive in managed hives.
This is what makes Australia paradise for the honeybees. Wild hives like this one, our pollination depends on it. But if the Varroa mite should ever invade Australia, wild hives like this will be the first to die.
So they would collapse first. And by collapsing, the Varroa mite populations will then descend on the commercial populations and the management becomes more difficult.
Queen bee breeder Tiffany Bates manages the research hives at the University of WA.
For bees, the Varroa mite is the big scary. And once that arrives here, we'll have about 3 to 5 years before the massive collapse of potentially commercial beekeeping. They can feed themselves pretty well, but
the question is, can we breed a commercial bee population in Australia that's resistant to Varroa mite? Today, male drones from the uni bee yard end up in the lab as sperm donors.
So, here comes the sperm.
Each male has this elaborate apparatus for docking with the queen in flight. Known as a suicidal mater, he blasts a sperm package and his penis into the queen, an ejaculation so violent it kills him. Undaunted, males take flight every afternoon to search for a queen by following her scent. To collect drones in the area, Tiffany baits the net with just a tiny amount of queen pheromone.
It's enough to have drones from feeding on all the trees around here and probably the bee farm just over there, flocking in. It doesn't take them very long at all. It's a remarkable demonstration of not only their ability to sense incredibly well, but to navigate really keenly.
so in theory, unless it's only our males, there should be a bit of a mixture in here.
In Australia, we're still fortunate to have plenty of healthy wild bees mating with hive bees, so there's constant genetic exchange. Amazingly, after mating with up to 15 males in a flight, the queen somehow selects the sperm she wants to keep. This tiny organ called the spermatheca holds the future of the entire colony. With it, the queen fertilises up to 2,000 eggs a day for the rest of her life, choosing from the sperm stored inside.
One of the questions is, of course, what does she want? And I don't yet know what she wants.
While the male dies after only 3 weeks, his sperm lives on in the queen for another 7 years.
It's easier in the spring.
Boris Bayer is artificially inseminating virgin queens to find out how they do it.
The secret of long-term sperm storage can be compared to the fountain of youth. The female provides a major liquid, and that does it. A single substance that we think is responsible to kind of turn sperm on, like really poof.
And the sperm itself triggers changes in the queen's brain that enable her to control the hive. Unravelling how the queen manages the genetic diversity of the colony is crucial to understanding how bees can resist Varroa. For example, worker bees help clear the hive of parasites and diseases by removing dead or diseased individuals. This hygienic behaviour is inherited, and bees that are good at it have a better chance of standing up to Varroa.
So if we can select for these traits, in particular hygienic behaviour and things like that, then we can try and breed bees that are more resistant. But at the same time, we need to really be careful that we don't breed out the genetic diversity that's present in bees.
Molecular biologist Matt Welch is delving deep into the honeybee genome to explore the interplay between brains, genes and reproduction. He studies the biochemical reactions that switch different genes on and off. This process, called epigenetics, doesn't change the actual DNA code, but how genes are expressed. To extract DNA from queen bees, you need fresh brains.
So queens have a much smaller brain than workers because the workers have a lot more sensory information to process. This is the part that we're really interested in, this central portion of the brain.
No larger than the head of a pin, it could hold the key to switching on a defence against Varroa. To find that key, powerful next-generation sequencers can rapidly read billions of genes from honeybee DNA.
So this has really unlocked our ability to do whole genome studies and to really explore genetic regulation and gene expression.
Varroa mites were able to jump from their Asian host to European honeybees by recognising their chemical messages as signals to lay eggs in their hives. It just might be a chink in the mite's armour.
If we can find a way to mask or block either the receptor that detects that signal or even the genes that encode that signal in honeybees, we could potentially make the European honeybee invisible to Varroa mites again, effectively eliminating their reproductive cycle.
Like a vaccine?
Yeah, that's right.
That prospect where honeybees escape Varroa's life cycle is an exciting one. But in the meantime, there are some other more fundamental mysteries to solve.
Swarms of bees operate in a vast volume of time and space, but nobody really knows where they go. Now, for the first time, miniaturised technology offers the opportunity to monitor their environment from a bee's point of view.
We give each one of those bees a badge, and we follow them. We give each one of those bees a number.
Physicist Paulo de Souza is working with entomologist Steve Quarrell to attach tiny sensors to the backs of middle-aged worker bees.
We're trying to get bees that are right on the edge of starting to fly, but we're trying to get the full flight duration of the bee, so 2 to 3 weeks. The hairier they are, the younger they are. So we're after, well, bald bees almost.
The tags on the bees are like pay pass on your credit card. A reader at the entry to the hive clocks them in and out. It's fiddly, painstaking work.
Next spring, when we do the full experiment, it'll be about 50 bees per hive per day over a 4 or 5 week period.
So in total, at the end of the project?
About 5,000.
Their first experiment will see if neonicotinoid pesticides have any impact on the bees' activity. These chemicals have been banned in Europe for 2 years because of concerns they affect bees' navigation, reproduction and immune responses.
So normally benign viruses and diseases or mites, say Varroa mite, are having a greater impact in the hive than was previously thought, basically due to these trace quantities of pesticide.
They plan to put traces of pesticide in feeder stations with tag readers to detect any changes in flight time and feeding behaviour. But the swarm sensing should also be able to tell if pests start turning up.
So we know if a new species arriving would drive any changes in the hive, would drive any changes in the ecosystem, and how that will affect our industry. We can transform the way we do biosecurity, and this is what we want to achieve.
John Evans is hosting the research on his apple orchard. He hopes to use the information to position his hives for the best pollination.
There's things about bees that we don't know. I don't think we probably spend enough time in understanding where bees are and what they do because they're so beneficial to us, and there goes one now.
The same is true for urban environments. Doug Purdy keeps high-rise hives in Sydney.
I think having beehives in the city is immensely valuable. Clearly we don't have a lot of pollinators in the city areas. Because of our fear, we tend to remove them. So putting backyard beehives in has got to be a good thing.
If people are wondering what they can do personally to help the bees, what would you recommend?
Look, it's easy for people to help bees. If you look at what we're planting in the gardens, people love grass and things that don't flower. Well, all they have to do is start planting things that flower. Because flowers are what bees need. You've got flowering things, bees will come.
If we want to keep taking advantage of all the benefits that bees offer, then it's only fair that we provide good food, shelter and healthcare in return. Because in the end, if it's good for bees, it's good for us.
There's something about the sound of bees on a summer afternoon that's just deep contentment. And I think that's why I'm here. Apart from all the bees are amazing in pollination and all of those things, I'm in it because when it works with absolute perfection.
How much water should you drink? If you've heard the hype, you'd probably say 8 glasses a day. But is it true?
No. In a single word, no.
There really is no evidence to support that view. So it seems very unlikely that this notional 8 glasses of water has any basis in fact.
So which areas are you looking for, Michael?
Well,
indeed, a study conducted by neuroscientist Dr. Michael Farrell suggests you can drop the mantra of 8 a day and rely on your thirst instead.
We were interested in knowing how the brain responds when people drink water that they want, when they're thirsty, and we contrasted that with drinking water when they were not thirsty, when they were drinking water that was in excess of wants and needs.
The study required a hard-earned thirst, so the participants exercised for an hour, sweating out 1% of their body weight.
My lips are pretty dry. I've been licking them for the last 10 minutes or so.
As we sweat and we're losing water but hanging on to salt, generally the level of saltiness of the blood increases, so too the saltiness of the fluids around cells, and this is the signal that is picked up by the osmoreceptive regions in the brain and in turn generates the sensation of thirst.
Our osmoreceptors in our brains can pick up a 1 to 2% change in the salt concentration of your blood and body fluids, so it's very sensitive.
Here we go. An MRI analysed the patient's brain activity as their thirst was quenched.
We simply use a syringe and some tubing to squirt small volumes of water into his mouth periodically, and we're very interested in what happens in the brain at that moment when he's swallowing.
That water really hit the spot. Yeah, really very nice.
As you drink, this region of the cingulate cortex, the pregenual cingulate, becomes activated, and we interpret this as representing that pleasant sensation you experience upon sipping water when you're thirsty.
But that changed when the participants were given more water than they needed to simply satisfy their thirst.
Then the sensation of drinking water becomes unpleasant, and that unpleasantness is associated with activity in a distinct region of the cingulate cortex, the mid-cingulate cortex, that we can see here in blue.
This unpleasantness also appeared to activate the periaqueductal grey, or PAG. It's a primitive region that's been shown to inhibit swallowing in animals. So with the PAG saying stop swallowing, it seemed the motor cortex, a more conscious region of the brain, was needed to override the PAG and help kickstart the swallowing.
Yeah, certainly it goes from something that you want to do and comes very naturally, that swallowing action, to something you really have to think about.
We are interpreting our results as a mechanism that protects us against excessive drinking of water.
In fact, it's possible to overhydrate. In very rare cases, drinking too much water has been fatal. Even excessive drinking during intense exercise carries risk.
The brain will put in place mechanisms to help the cells in the brain hang on to their water as the water outside the cells becomes saltier. This mechanism isn't turned on and off quickly, so if a great volume of water is ingested, this is when the risk occurs of swelling and potentially death.
The crucial thing about this water intoxication is the rapidity at which you ingest the water. If it's done very slowly, it's probably not going to have very adverse effects, but if it happens rapidly, then it can be very dangerous.
The fact is, you get around 20 to 40% of the water you need each day simply from the food you eat. And even moderate tea and coffee intake will hydrate you, as their diuretic qualities are quite low. But the same can't be said for alcohol. So you can trust your thirst, unless you're 70 or over. While older men had levels of thirst similar to younger men, it was found they drank only half as much in response. It seems their thirst mechanism turned off too soon.
They were getting enough to satiate their thirst, but they were not getting enough to rehydrate them.
That puts the elderly at greater risk of dehydration in hot weather.
If you want to make sure that you're drinking the right amount of fluid, probably an easy way to do it would be to weigh yourself in the morning and then make sure you don't drop too far below that body weight.
And in case you're wondering, excessive amounts of water won't flush out your toxins. After reviewing the science, Kidney Health Australia rejects the target of 8 glasses a day.
Drinking 8 glasses of water may be well in excess of need on some days and totally inadequate on others. So it doesn't reflect the reality of how we live.
We all know texting on a mobile phone while driving is a very bad idea.
So I'll meet you out the front entrance
But
at about
what
8:30.
about hands-free conversations?
Okay, thanks, bye. Although it's illegal to hold my mobile phone and drive, hands-free conversations are still allowed. My eyes are on the road and my hands are on the wheel, so it's perfectly safe, right?
I've come to the Queensland University of Technology, where Professor Simon Washington is looking at this question in detail.
So when we put people in the simulator, we can measure everything they do in the car. We can measure when they brake, we can actually monitor where they look, as well as when they react to certain obstacles or events on the road. And in this particular study, we're looking at young drivers and trying to understand the effect of distraction on what they do in the car.
Now it's my turn.
Thanks.
I'll be taking a drive test in 3 conditions: without a phone, talking hands-free, and with handheld.
You just drive as you normally do.
Pedal to the metal.
Yeah, okay.
Okay.
Okay, have a safe drive.
Thanks very much. And do I use my indicators? All right. Oh, this is really strange. This is bizarre.
It's a 40km/h speed limit, so no risk of a high-speed smash. But there are plenty of potential hazards like stoplights, cyclists, and busy intersections. It's a strange sensation, but on my phone-free test, I negotiate the course without any mishaps. Then Shamal makes a call.
Hello?
Hello. Jane is driving faster than Kim, who is slower.
Shamal is asking me to solve simple problems which increase my mental load.
Jane, it's Kim.
It's soon clear that it's only possible to give complete attention to either the conversation or the driving, but not both.
Undetected by the sleeping dog, the thief broke Jane's apartment. What was the dog doing?
I don't know. I didn't listen. I was making the turn.
Undetected
Am I going the wrong way?
On hands-free and handheld, I miss signs to the airport, miss time traffic, and pedestrians seem to come out of nowhere.
Sorry, I just nearly hit a pedestrian. What did you say? Sleeping.
What happened in my test mirrored the results of the trial. Phone conversations made younger drivers less aware of what was happening in their peripheral vision.
What we found is when something is happening right in front of you, there's almost no distraction. There's almost no increase in reaction time. But if it's to the side, those kinds of events that in the peripheral vision are very, very difficult for someone who's distracted to detect. What we find is there's about a 40 to 50% increase in the reaction time.
This means if you take 2 seconds to notice a pedestrian coming from the side, it takes 3 seconds when you're on the phone. At 60 kilometres an hour, 1 extra second takes your car 16 metres closer to the crossing. And it wasn't holding the phone that was the dangerous bit.
There isn't that much difference between hands-held and hands-free conversations. That the cognitive engagement of someone is the most important aspect of the distraction.
Brain scans taken in another drive simulator study shows exactly how we lose focus. Normal driving engages the posterior part of the brain, which controls visual and spatial awareness. But when a conversation takes place, activity shifts to the prefrontal cortex, which controls decision-making.
And so in a sense you get tunnel vision that comes in like this, and the parts of the brain that are responsible for seeing things out here are diverted to the phone conversation.
But we can't stop having conversations in cars.
So we're having a conversation right now. Your study seems to suggest that that's a very dangerous thing to do, that I'm not going to notice things as much in my periphery.
Well, actually, you still will notice everything in your periphery. It's because as a passenger here, I might point things out to you, and I'm not going to overload you with questions and discussion when there's a high-risk situation coming up for you as a driver. Turn right here.
But really, that all depends on the passenger. By analysing families on real trips, the Accident Research Centre at Monash University are looking at just how much distraction children can provide. It's a lot.
What we found was that children were 12 times more frequent in their capacity to distract the driver than were cell phones. We saw drivers engage with their children in a variety of ways, including reaching back to pass them food. Of course, we see conversations and singing and so on. In an average 16-minute trip, the parents were taking their eyes off the road for around 3 minutes and 16 seconds. And this clearly is of concern.
It may not be feasible to ban either kids or mobiles from cars, but realising the danger of buckling to their every demand will go a long way to protecting the ones we love the most.
Next time on Catalyst, mega tsunamis. Groundbreaking Japanese research reveals new clues about future hazards for tsunami-prone countries. Could the bacteria found to cause stomach ulcers help cure allergies? And outdoor play, how it can improve children's mental and physical health. Connect with Catalyst on Facebook, Twitter, Instagram, YouTube and our website.